This video helps us understand laboratory number 1 on the scientific method. The purpose of this lab is to learn the steps of the scientific method, learn the parts of a good experiment, and be able to identify controls and variables in experiments. There is no single rigid method that all scientists use. However, there are similar investigative processes. The steps of the scientific method include observation, questions that arise from these observations being the things you might see, hear, or otherwise take in with your senses. We form a hypothesis, a tentative explanation for the phenomenons we observe. That word comes from Greek root words, hypo meaning under, and thesis meaning argument. The hypothesis is the basis of a scientific statement or argument. Then we make predictions based on our hypothesis. We apply it to an experimental situation. The logic involved in a prediction can be expressed as a conditional statement. We say, if this happens, then that will happen. The hypothesis is stated as if-then statements to determine scientific truth, to determine what parts of the world cause certain effects in other parts of the world. Then we test the hypothesis through our experiment and record the results. We spend time mathematically analyzing those results. We use statistics for that. And then we go back to our hypothesis and conclude if the experiment supported or refuted it. From there, if it was a worthwhile experiment, we communicate the results by publishing our scientific experiment in what are called scientific journals. In many fields such as science, proof does not really exist. The only thing we have are observations, facts, and recorded data. I'm using proof in a very specific scientific way here. We ask ourselves, does the data of the experiment support or refute the hypothesis? If it refutes it, we need to change the hypothesis. Hypotheses often change after experiments suggest the truth is something different. Nothing in science is proven forever and ever. All we have is tentative proof. The scientific truth can change if a new experiment shows that it needs to be modified. Tentative, meaning that things are only true for now. So how do we use science then? There's different kinds of logic or reasoning that comes from the scientific endeavor. Inductive reasoning is when many specific observations lead to a general idea. For example, you do many experiments and it leads to a theory of gravity. Once we have a working theory of gravity, we can do what's called deductive reasoning. That's using a general idea to apply the theory of gravity to specific situations. For example, we might say if gravity is a good theory, then we should design a plane like this. So we take our ideas and we apply them to things. The different parts of an experiment require careful design. We have to isolate the different variables. This helps us determine the relationship between the cause and effect in the world. What causes certain things? Weak experiments fail to clearly show the effects of one variable. For example, herbal supplements are different from the kind of medicine that needs prescriptions because the exact chemical involved in an herbal supplement is not clear. When you get an herbal supplement, you get the whole plant. When you get a medicine like aspirin, there is a specific chemical that has been tested to show a specific result. Some situations have many variables, things like human intelligence. Many things in the world act in complicated ways. That can make experimentation difficult. Next, we can see that advances in technology can change experiments. They change how we can test the world. For example, it wasn't until the medieval era that glassware was advanced enough to handle the pressure of intense heat or be able to magnify things. So glassware was a technology that opened up parts of the world to us. Likewise, computers have opened up all different kinds of things, and then the internet beyond computers. We can look at other parts of an experiment. A good experiment uses variables, and that means the things that you can change in an experiment and might influence an outcome. We have specific kinds of variables. Controlled variables are those factors that could change but are prevented from changing during the course of an experiment. Controlled variables are things kept the same. Keeping things the same allows us to see that there was one specific thing that we changed. So controlling variables helps us isolate the one that's really important to us. Then we have variables called the independent or experimental variable. These are the variable that impact or affect the outcome of the experimental situation that the scientists set up. These are variables that scientists can control. So for example, you could give a group of people aspirin daily, and another group of people you tell them to live their life. The presence of the medicine, the aspirin, is independent. The scientists could control that by telling the people what to do. And then you could watch who has heart attacks over many years of having aspirin daily. The scientists can't control who gets heart attacks. They're not in there touching the person's heart. All they could control was whether or not they took aspirin daily. Or you could use different fertilizers, an independent variable, to measure plant growth, a dependent variable. The scientists can't directly control the plants growing larger. What they can control is which kind of fertilizers are used. So the independent variable is the thing that the scientists control. Then the independent variable's values are controlled or selected by the experimenter, as I said. This way, it helps reveal relationships to the responding or dependent variable. The dependent variable is the event studied and expected to change as a result or in response to the independent variable. Independent means standing on your own. The dependent means it relies on or depends on the different kinds of independent variables. So those are the things the scientists try to observe. Those are the things that they honestly care about to make the world a different place and make it better. These are the results they're looking for. Preventing heart attacks or getting plants to grow larger. Those are dependent variables. The scientists don't have loyalty to aspirin or one brand of fertilizer. What they want is people to have better health or more food. Going on. There are different groups that participate in an experiment. One group, called the control group, will get variables that are controlled for or held constant. This group of participants in an experiment have situations most similar to normal conditions. Normally, people wouldn't get aspirin. That's not a thing that they go after daily. So that's a control group, people just living their lives. On the other hand, the experimental group is the group in which one factor or treatment, that's the independent variable, is varied. They get something different. They get medicine. Or some plants get fertilizer, and other plants just get regular old dirt. That would be the control group. People taking aspirin would be the example of the experimental group in the one example I've been talking about. The larger the groups you get, the more it helps with statistics to show that there's an actual effect of the variable that the scientists are manipulating. Here we see another example. If the scientist asked, will student test scores be affected by distracting sounds in a testing environment, a control group would get the situation that is most normal, quietly taking a test. The experimental group would have the independent variable introduced, a band, playing distracting sounds, and then the scientists would measure the dependent variable, the results of the test.